Hi, my name is Pavi, and in this video I'll be introducing you to the mTOR pathway, how it's activated by leucine, and the many major implications that it has for diseases such as cancer. To start, cells are incredibly complex organisms that need to be able to adapt and respond to their environment. This means being able to respond to changes in nutrients, energy, and stress. The mTOR pathway is responsible for integrating all of these complex inputs. At the center of this pathway is the protein mTOR, or mammalian target of rapamycin. mTOR is a kinase, meaning that it catalyzes the transfer of phosphate groups onto substrates like other proteins. The mTOR protein forms complexes with various other proteins in cells. The most well-studied is mTORC1. This complex responds to many different inputs in order to drive cell growth, mainly by activating other proteins in the cell, such as ones that control translation of genes and regulation of pathways, as shown in the figure. Because of this, mTORC1 is known as a master controller. Research has shown mTORC1 to be linked to diabetes, cancer, and even aging. Mutations in mTOR regulators have been shown to drive tumor formation, and are also prevalent in human cancers. So, understanding this fundamental pathway is incredibly important. Research has shown that mTORC1 integrates information to regulate growth. Nutrients like amino acids and glucose, growth factors produced in the body like insulin, and energy availability through ATP levels all affect the mTORC1 pathway. Environmental stresses like DNA damage or low oxygen levels can also signal to mTORC1. Out of all of these inputs, though, the most important is amino acids. Studies have shown that if amino acids aren't available to cells, even an increase in the other stimulants like growth factors or stress won't stimulate mTORC1. Why are amino acids necessary, and how do they activate mTORC1? Well, research has shown that mTORC1 is directly activated by a protein called Reb that is found in the membrane of lysosomes. In order for this activation to happen, though, mTORC1 and Reb need to be close to one another on the lysosome, but usually mTORC1 is floating around in the cytosol. Experiments show that a complex of proteins, called the regulator RAG complex, is also found in the lysosomal membrane. The RAG proteins in this complex can bind free-floating mTORC1, and thus bring it close to Reb for activation. Amino acids are somehow necessary for RAG proteins to capture and bind mTORC1. The most well-studied amino acids are leucine and arginine, both of which are known to stimulate mTOR signaling. Recent work in the field has helped uncover exactly how leucine promotes mTORC1 activation and mTOR signaling. After eating food, say a piece of chicken, the proteins within are digested by the body into amino acids, as shown by the blue dots in the figure. These amino acids are then absorbed into the bloodstream and transported to cells around the body, where they are absorbed through specific transport proteins. Recent research shows that once leucine is inside the cytosol of the cell, it is sensed by a protein called cestrin-2, whose molecular structure is shown here. Cestrin-2 has a pocket for leucine to bind. The base of this pocket is hydrophobic, and forms van der Waals interactions with the hydrophobic side chain of leucine. In the second figure are the charged glutamic acid and arginine groups, which anchor the leucine by interacting with its amino and carboxyl terminals. The hydrogen bonds are shown with black dotted lines. A loop of amino acids from cestrin-2 forms a lid on top of the bound leucine, as shown in the first figure. This loop actually hydrogen bonds with another part of cestrin-2, forming a latch to hold leucine in place. In the figures, these hydrogen bonds are represented by black dotted lines. How does cestrin-2 binding leucine lead to mTORC1 activation? Well, cestrin-2 interacts with other proteins, which interact with other proteins in a complex manner that isn't entirely known. The end result, basically, is that the RAG proteins in the regulator RAG complex become active, and can bind mTORC1 from the cytosol and bring it close to Reb. mTORC1 is then activated and goes on to regulate many other proteins and cause numerous effects in the cell. These effects are incredibly complex and are still being studied in detail today. Inhibiting mTORC1 in the mTOR pathway has been found, for example, to elongate lifespan in mice. mTORC1 activation is also necessary for protein synthesis and building muscle, such as after a workout. Studies have also been finding links between mTOR and diabetes, Alzheimer's, Parkinson's, and many more diseases that can't be covered in the length of this video. One area of especial promise, though, is cancer. Knowing how amino acids activate the mTOR pathway can be useful for the treatment of cancer. One study of prostate cancer in mice found that inhibiting amino acid transport suppressed mTORC1 activation. Furthermore, when these mice were injected with cells derived from human prostate cancer tumors, they found that after 29 days, mice in which amino acid transport had been inhibited had decreased tumor growth. This is shown in the highlighted boxes in the figure. Furthermore, these mice also had a reduced overall metastasis of these tumor cells. The mTOR pathway is thus a promising avenue of treatment for cancer. but much more research will need to be done going into the future. Much is still unknown about the exact mechanism by which amino acids stimulate mTOR1 activation, as well as the many other possible stimulants of the mTOR pathway. Also, much is unknown about the downstream effects of the mTOR pathway and its involvement in many human diseases. Hopefully, future research will be able to address these important questions.